This was a more difficult piece of research than I had thought. And this is part of the reason why I've started a little bit late, because I was trying to piece an idea together. This is what happens. Comes in front of me, and I try and make sense of it. And what happened today, so that you understand, if you can, highlighted some work he was doing looking at the spike protein and amyloidosis. And he sent quite a comprehensive document for me to look through, and I was going through it, and there were just some questions that popped into my head, so I'm sharing with you some ideas. So before I go any further, it's important for those people who will be trying to criticize the questions. These are just ideas that I am talking about, and I think that they could be relevant in the context as to what it is that we're looking at. So, my question is, why do these strange embalmer's clots occur? So, I'm not even going from the perspective that people think this is a conspiracy. No, it's not. It's real. They've studied it, they have looked at the characteristics of it, and we don't know why it's occurring, but it has some very unusual patterns that need to be addressed. One of them is that it has a high concentration of tin. Now, tin has no biological function in the body, and it could only get in the body from outside. But even if it did, why in the world would it be in a clot? And this is part of the question that triggered the sequence of my thinking. And this is what took me longer than I had thought about. And I was then trying to think, okay, the most unusual thing that I've seen about these clots is that tin is in a concentration that you would never expect in it. So I then started to relax and think and work my way through the potentials, the possibilities as to how in the world tin first would get in the body and second be able to be concentrated in the clots. So I am starting here from the perspective as to understanding what is going on, and it is just a theory. Before I start, just a reminder, coming up this weekend is this conference, NeuroCovid, and the neurological aftermath after COVID-19. Tremendous speakers and panelists are present here. You really want to join us. It will be live and available, but for those people who don't want to forget, make sure you register and you will get the link and the reminder. Plus, I'll be able to see the people who have registered, and I will try my best to prioritize your questions. So, getting back to this strange pattern with regards to these clots, let's start with a few basic things. Embalmer's clots are real. This is Richard Hirschman with one of them, a 19-inch clot that was taken out of somebody who died. Part of the problem is that the scientists who are debunking it are suggesting that this could happen post-mortem, and so therefore it's not as relevant. However, I have got clots from the whistleblower who sent images of clots. Pity I don't have it in my my description below, but if you go to my Substack and just type in whistleblower and search it, it will appear. And so we know it's occurring not just in dead bodies, but in live people. We need to study it in live people as well. So one of the unusual patterns is that it has a number of unusual proteins in this clot. Everything in red is not normal. In black is normal. So you can see fibrinogen beta chain, gamma chain, alpha chain, but you don't normally see hemoglobin in the alpha and beta units, immunoglobulins that suggest that there's some kind of immune response, myeloperoxidase, cathepsin G. All of these things are not normally in a clot. So we know that there is something abnormal that is happening. But when you look at the characteristics of the clot, it is so well formed and amyloid, meaning that it's like a crystal and it grows. And so this doesn't happen quickly. And so therefore, even more unusual that tin is incorporated into this. So this is the perspective that I'm going from, the tin question. Some of the things that, based on the research before that I had looked at that I thought was relevant, is the main constituents in terms of the proteins in it. I certainly believe that neutrophil and macrophage nets that are released to bind properly spike protein or otherwise, definitely spike protein with heparan sulfate, platelets, activated platelets, and their products, and spike protein with hemoglobin. This, from my research, is the main constituents that contribute to this very well-formed gelatinous amyloid clot that cannot be dissolved. So it's very unusual in that sense. When the whistleblower spoke about it, he clearly indicated that from what he could remember, and he couldn't formally go through all the notes, but the large majority of people were vaccinated. Now, again, that's not formally corroborated, but it would make sense that if it is in some way connected, we don't seem to be hearing about it in low vaccinated countries. Now, whether or not they're just not doing autopsies or they're not looking, I don't know. But his view was that it was somehow connected to the vaccine. And I have always thought it was more likely to be an immune response triggering this kind of complex mechanism and not necessarily connected. But the question of tin really bugs me because something about it doesn't make sense. So here is the hypothesis. So the first thing that you have to realize is that this is a lipid nanoparticle. And what it is really a ball of lipids and cholesterol and so on, stabilized with the mRNA in it. Because if the mRNA was exposed, it would get destroyed quite quickly. And so the mRNA is put in this package, which is then injected and delivered inside cells, which then makes the spike protein. And that's the principle of the mRNA vaccines. Similar, slightly difference with the adenovirus protein-based vaccines, just inject them in. Now, one of the problems is that people have become so focused on the spike protein and the mRNA that they may not have clued in to other pieces of the puzzle. And so hear me out now. This is where I think I'm starting to wonder. When you look at the lipid nanoparticle itself, okay? And in this picture, I got this from Inside Therapeutics. I thought it was a really good image. So this lipid nanoparticle still has the mRNA in it. However, it has around it these ionizable lipids. These are not natural. It has as well on the outside of it phospholipids, it has cholesterol, and it has these PEG lipids. These are also uniquely created in order to stabilize this whole structure. And it's this that is taken up inside the cell, then broken down to release the mRNA, which then makes the spike protein. So the question becomes, what happens to the rest of the stuff? And the way that I think about it is similar to what we're having a problem with plastics in the ocean, is that in effect, after you do all your shopping and you have all this plastic, it's not biodegradable. And this is why we have a problem. And so the question with regards to the lipid nanoparticle is which parts are easily biodegradable? And when you look at it, the bits that will get degraded easily and incorporated are cholesterol, the phospholipids, the RNA will get destroyed, The PEG lipid, to some extent, is not so easily digested, but this ionizable lipid may be the hardest to break down, okay? Because this is not a natural product. And so what you'll be left with is these and these, somewhat kind of like the plastic bag floating around in the ocean. So this is now where my theory goes. Let's work with me here. So my theory, and this is just based on me doing some research, took me an hour and a half of just thinking and questioning and thinking, and this is what I've come up with. I think it is possible that tin has become embedded in the lipid nanoparticle. And this is pre-injection. And because I cannot find any other way, so tin is SN, and in effect, I'm saying that it's stuck in here, and actually it's probably most likely related to the non-digestible ionizable lipids. And someone will say, well, there is no evidence that tin is in the lipid nanoparticles or even in the vaccine because they have tested it and they haven't found it. Okay? Hear me out. So as I looked at this, as usual, invariably, there's going to be somebody who is doing other research. And in this paper here, this is ResearchGate, and they don't like this paper, so it's gotten pushed away. But they claim that there are at least 55 undeclared chemical elements found in the variety of all these vaccines. They have done their own independent research, and it has been debunked because they say that it's not been standardized. They don't know the source of where they have been getting the testing from, because it certainly didn't come from the pharma industry. And so I don't know where they got the vials from, all of these questions. And so largely it has been ignored. But for the purpose of what I am looking for, they have clearly identified a number of things, including tin. And this is just highlighting the isotopes here. And they looked at which one of the vials, and they were looking at certain Pfizer vials here that had it present in it. And so it seems that it is possible that it occurs. And my thought is that the reason that the tin may not have been found in the previous testing, because they probably did look at it, is that when they tested it, they may not have captured it in free floating fluid, because it is embedded in the lipid nanoparticle. That's my theory. I am not a chemist, so I can't absolutely confirm this. My problem is that I am trying to figure out how tin gets embedded in these embalmer's clots. So based on that theory, let's assume for a moment that my theory is correct, that tin somehow, whether it's from the you have to put a plug in these valves. It's not necessarily in the manufacturing process. It could be related to just the packaging of the product, and so therefore it wouldn't have been found if you, depending on where it was tested for. And so it's not a lot of tin, but it's enough to get caught up in lipid nanoparticles. And if you remember the principle, it then becomes like plastic bags with tin on it. And what I am then proposing is that these remaining lipid nanoparticles, now getting rid of it, the best scavengers are these macrophages. And the macrophages, I would predict, would munch up anything like that and pick it up. But the problem would be is that it can't digest it, because they're not really digestible. And so it could get stuck inside these macrophages. And then in the context of what happens, because it then would have to become embedded in proteins within the macrophage, because the macrophage would also be picking up spike protein, all kind of rubbish that's lying around or floating around. And you could then have a situation where almost a protein is formed composed of multiple things inside the macrophage. So then you may ask, well, how is it that it would get in the clots? And this is now would come back to my original theory that these clots are immune mediated. I think that they are triggered in some people on exposure, re-exposure to the virus. Let's assume they already have lots of these pieces of plastic stuck in macrophages. And critically, remember I told you that macrophage netosis, where the macrophage releases these nets to trap other spike protein, maybe virus, whatever, then becomes a huge conglomerate, in my mind, looking something like this. So you have the macrophage here, another macrophage here. They're releasing these nets. It's trapping spike protein, immunoglobulins, everything else. And in theory, then, also caught up in these nets could be the pieces of the lipid nanoparticles, which would then contain tin. And then when this starts to grow because of the continuous process, you then end up with a very solid protein that's not easily digestible, but having a high concentration of tin in it. That's my theory, because I can't find any other logical explanation for where it has come from. Because why would some people just randomly have elevated levels of tin in their serum? But it's not serum, it's incorporated into protein. So it's not free floating. Free floating would then get excreted, but it would then have to be connected with maybe fibrinogen or hemoglobin in order to get into these very unusual clots. And this is what they would kind of look like. This is what the embalmer's clots look like, is that these white fibrous, clots, different from the dark clots. This is the kind of thing that they are finding. I find it remarkable that I've been speaking about this for over 2 years, and there is so little interest to investigate it. I think that in another 50 years, when they look back, that is going to be one of those moments where the scientific community in 50 years' time says, I don't understand. If this was happening, why didn't you investigate it? Forget about whether or not you think, well, it can't be a conspiracy because it's real. Nobody has really seen it before. Why didn't you investigate it? What reason, what logical reason could there be if there is a pattern that is occurring that has not been seen, that initially was debunked as unreal, but the longer it's going on, and they have now been, it's not even as if you can say that you disagree with it, because this has now been looked at in detail under the electron microscope. They've taken the constituents of the clot. They've found that it has high level of tin. It has an amyloid appearance. I mean, everything about it is indicating this is something very, very unusual. And yet, there is still no serious interest to investigate it. What can I say? I hope, therefore, in 50 years' time, they will say to me, thank you for asking the questions, because somebody had to. But we'll keep working at this, and hopefully one day it will become something that is taken seriously enough that they actually do research on it. As a final point, this is my hypothesis. I'm just sharing it with you. So in case the sensors are floating around, it's just an idea. There is no evidence that this is happening. This is just me extrapolating the thought to explain why tin could be involved in this. And please remember to like, subscribe, and comment. It helps to spread the news around. Share your ideas. Let's see if we can come up with a better and more solid scientific solution. Have a great evening.